Today we're taking a closer look at a really fascinating topic in pharmacology. We're talking about how certain molecules, these things called antagonists, actually work. We've been digging into some material that, lays out the basics and gives some pretty specific examples too.
That's right. And our goal here for this deep dive is really to unpack this material. We want to understand what agonists and antagonists are, how they actually interact, at that fundamental level. And then we'll get into the different types, specifically competitive antagonists, and really explore why they're so important in medicine and research as well. It's kind of about understanding the molecular dance happening inside your body and how we can, influence it.
Okay, let's unpack this then. We probably need to start with the basics, right? What exactly is an agonist?
Right. So think of an agonist as like a drug or even a natural substance your body makes, maybe a neurotransmitter or a hormone. It finds a specific spot on a cell, usually a receptor, and it binds there. And the key thing is when it binds, it activates that receptor.
Activates it, like flipping a switch.
Yeah, exactly. It's like a key fitting into a lock and actually turning it. That turn kicks off a whole chain reaction inside the cell, which leads to some kind of specific biological response.
Okay, got it. Agonist equals activation. So now, the main focus for us today, the antagonist. How's that different?
Well, antagonists also bind to those same receptors. They find the same spot.
Yeah.
But here's the crucial difference. They don't activate them.
Okay. So they fit in the lock, but they don't turn the key. Is that right?
That's a perfect way to think about it, yes. And the material we looked at really highlights why this is so important. For researchers, antagonists are like vital tools. They help figure out what specific receptors actually do.
How so?
Well, if you block a receptor with an antagonist and then some specific function in the body stops, you learn that the receptor was probably involved in that function, right?
Makes sense.
And then clinically, in medicine, they're used all the time. You use them to block receptor activity you don't want, or maybe to counteract the effects of an agonist that's causing problems.
Like turning down a signal that's too loud, maybe?
Precisely. They act as modulators. Really important ones for regulating body processes for therapeutic benefit.
Okay, so how does this blocking actually happen? Let's try and picture this interaction. You've got the receptor on the cell surface. It's got a specific shape.
Right. Shaped perfectly to bind with its natural agonist and cause a response.
And when the agonist is there by itself, it binds, activates, and you get the normal response. Simple enough.
Yes. But now let's introduce an antagonist into the picture. The antagonist, it usually has a shape that's pretty similar to the agonist. Similar enough anyway. So it can also bind to that same receptor site.
Okay.
But, and this is the key, it binds without activating anything.
Right. The key fits but doesn't turn.
Exactly. And this is where the competition aspect comes in. It's not passive. The antagonist physically gets in the way. It occupies the binding site.
So the agonist can't bind there anymore.
Not if the antagonist is sitting there. They essentially compete for the exact same spot on the receptor.
Yeah.
By occupying that site without doing anything, the antagonist hinders or, antagonizes the agonist action. It reduces or sometimes completely blocks the response you normally expect from the agonist.
It's like someone taking the last parking spot right in front of you and then just sitting in their car.
That's a great analogy.
Yeah.
Yes, they're occupying the space, but not performing the function associated with that space.
The material we looked at had some really good sort of real-world examples. This is where it clicks, I think.
Absolutely. Let's take antihistamines. We all know those.
Yeah.
Histamine, that's a natural substance your body releases, causes allergy symptoms, the itching, sneezing, watery eyes, all that.
Right, annoying stuff.
Well, antihistamines like phenyramine, cetirizine, loratadine, these are histamine H1 receptor antagonists.
So they go for the same H1 receptor spot that histamine wants.
Exactly. They compete with histamine for that binding site. When the antihistamine binds, it just sits there. It doesn't trigger the allergy symptoms. And because it's occupying the receptor, the histamine can't bind, or at least not as effectively.
So it blocks histamine's effects, reducing the symptoms. Okay, that makes sense.
Yeah, it prevents histamine from doing its job.
Okay, and then there's naloxone. This one sounds really dramatic. A powerful opioid receptor antagonist. The material says it can rapidly reverse opioid effects.
Yes, naloxone is incredibly important. It works against opioid agonists like morphine or fentanyl. In an overdose situation, these opioids can cause life-threatening respiratory failure, basically, stop someone breathing. Naloxone competes very, very effectively with those opioids for the opioid receptors.
It kicks some more.
It can displace them, yes. It binds to the receptor, blocking the opioid, and reverses those dangerous effects, especially the respiratory depression. It's literally a life-saving molecule in those situations, a molecule that steps in, takes over the receptor, and reverses the immediate danger.
Wow, that's powerful stuff.